Hi everybody, my name's Kathy Belov. Welcome to the Australian and New Zealand session of the meeting. Firstly, I'd like to acknowledge that no matter where we are in the world, we are sitting on the lands of Indigenous people. I myself am on Wallamedigal land of the Eora Nation, so that's between the Parramatta River and the Lane Cove River in Sydney and New South Wales. So I'd like to take a moment to acknowledge that Indigenous people have been working on this land and doing research on this land for many tens of thousands of years. And I acknowledge any Indigenous people that may be joining us today, their elders past, present and emerging. And we will see today that our working with Indigenous people is tremendously important to us in this region of the world. So today we've got 6 fantastic speakers, and you'll get a snapshot of the types of research that are occurring in Australia and New Zealand at the moment. It's just a short snapshot. Unfortunately, we don't have a lot of time. So what we're going to do is I'm going to introduce each speaker at a time. We're going to hear their talks, and then we're going to have about 10 to 15 minutes at the end of the session for questions. So please type in your questions as we go to make sure that you remember your questions for speakers, and also vote for questions so that I can have a look and see whether there are particular themes that are emerging. You may want to ask a similar question to each of our speakers, for instance, to get different perspectives on a view. So first of all, our first speaker is Anne McCartney. Anne is from Landcare Research in New Zealand, and she will be talking about the genomics Aotearoa project. It's been a long day for me, apologies. So she'll be talking about how she and her collaborators are working alongside Maori to better understand the biodiversity of New Zealand. Anne, over to you.
My name is Anne McCartney, and I was a postdoctoral research fellow for Genomics Aotearoa High Quality Genomes Project. And who are we? As Peter Dearden would have previously stated, Genomics Aotearoa is a collaborative research platform for genomics and bioinformatics. And what my role here was to work with collaborators to generate high quality genomes for species of national interest. And I also had the role of playing kaitiaki of these species. And kaitiaki in Maori, who are the indigenous people of New Zealand, means guardians of these species. And with this, I had to conduct all of my research in line with Maori traditions and cultural knowledge. And that went right back from project development and spanned right through to after the analyses had been completed and actually storing this data afterwards. So I want to talk to you today about how utilizing high quality genomes actually benefits your downstream analyses. And with this, I had 3 main projects that I wanted to talk about today. My first project that I want to talk about was to actually do a phylogenomic analysis of 25 endemic Phasmatodea species. And here I would like to acknowledge all of the iwi who I've listed here that contributed the samples and we worked alongside during this project. Why Phasmatodea or stick insects? They're actually really neat. They have a different climate tolerance as you span across New Zealand. They are parthenogenic, and they also form hybrid populations and hybrid zones. They have a cool and unique genomic structure. They are large, heterozygous, and repetitive as well. So they're very interesting. So in order to carry out the phylogeny of these Phasmatodea, we decided that we needed one single really high quality reference for the clade, and we selected Clitarchus hookeri. And we obtained chromium 10x, nanopore, and Hi-C data, and from that data we were able to scaffold this assembly onto 18 chromosomes. For the remaining 24 species, we had about 30x Illumina data. I carried out de novo spades assemblies from those, and with the program called Ragoo, I was able to then generate from my high quality reference-based assemblies for those low quality 24 other stick insects. From there, we were able to annotate all 25 as we obtained RNA-seq data. From there, again, we were able to then get our orthologs, and it was easy then to generate our species and gene trees. My second project that I wanted to talk about was the population genomics of Knightia excelsa. Knightia excelsa is a honeysuckle-producing Proteaceae plant, native and endemic to Northland, New Zealand. And again, I want to acknowledge the iwi that contributed the samples and that we worked alongside throughout this project. Why Knightia excelsa? As I said before, it's endemic to New Zealand, and it's of key primary production interest in the honey production industry in New Zealand. So before we conducted our population genomics of Knightia excelsa, we first needed to generate a really high quality reference for this species. To do that, we acquired Illumina, Nanopore, and Hi-C data, and we were able to scaffold that onto 14 chromosomes. In the interest of times, I won't go into the methods here, but if you have any questions, feel free to ask me at the end. With this really high quality reference assembly, it was then easy to carry out our variant detection pipeline across 34 pool seed populations that we sampled across Northland, New Zealand. The final project where we utilised these high quality genomes was to identify the toxin producing cluster in Pseudopithomyces chartarum. And we did this in collaboration with the University of Auckland and Ag Research. And why this is important is that Pseudopithomyces actually produces a toxin called sporidesmin A, and this presents itself in sheep and cattle as facial eczema, which is indicative of an underlying condition of liver disease in these species, and that actually causes a loss of about 100 million New Zealand dollars per annum in the dairy industry. So we decided to construct again a chromosome reference assembly for Pseudopithomyces, but we just needed Illumina and Hi-C in order to scaffold this onto 21 chromosomes. We then obtained 3 different strains of the fungi, 2 which were toxin producers and 1 which did not display evidence for toxin production. We assembled these genomes. We were then able to annotate and functionally compare all 3, and we found evidence for the gliotoxin and sporidesmin A gene clusters in the toxin producing fungi, but not in the non-toxin producing fungus strain. So I hope I've convinced you at this point that having actually a really high quality reference genome really benefits your downstream analyses. And with that, I would like to thank all of my collaborators on these projects, particularly the iwi involved and that we worked alongside throughout. And with that, I'll take any questions. Thank you.
Thank you, Anne. Next up, we have Paris Brandis. Paris is from the University of Sydney, and she'll be telling us about the reference genome of the Antechinus. And this is a great example of the value of comparative genomics for doing unique biomedical research. Over to you, Paris.
Hi everyone, my name is Paris and I'm a PhD student at the University of Sydney, and today I will be talking about the reference genome for the Antechinus, which is this little genus of marsupials that live in Australia and New Guinea. So the first big question is, why do we want to study the Antechinus? Well, Antechinus are a really interesting genus of organisms because they're one of the only mammalian organisms that exhibit this rare reproductive strategy known as semal parity. And this is when individuals only live long enough to reproduce once in their lifetime. So during a really intense breeding season, males will dedicate all of their resources into reproduction, causing a total immune system collapse and death shortly after mating. And so this means the Antechinus would be a really interesting model to exhibit the interplay between stress, reproduction, and immunity. In addition, Antechinus have also been found to develop age-related neuropathologies like those seen in humans, and have therefore been proposed as a good model for Alzheimer's disease as well. So an Antechinus reference genome would facilitate future research on these key interesting biological traits. So this is our project outline. We started with genome assembly using 10x linked read sequencing data. We then did a transcriptome assembly from a variety of tissues from both a male and a female Antechinus. We then annotated the genome using FGENAsh++, and then performed some downstream analyses on some key genes that we were interested in. These are our assembly results, and you can see that we use the brown antikinase as our target species, and this is because the brown antikinase is one of the most commonly studied antikinase species in research. The genome size was 3.3 gigs, which is what we expected based on other marsupials. The number of scaffolds was about 30,000, but the scaffold N50 was quite high, 72.7 megabases. So that means we had quite a few really long scaffolds in the genome, which was really good. The complete mammalian BUSCO score was 93.3%, again showing good completeness of the genome. The next step was then to assign chromosomes by aligning the antikinase genome to the closely related Tasmanian devil. And what you should see from this graph and table are that the chromosome lengths of the antikinase and the Tasmanian devil ended up being quite similar using this approach, which is what we expected because dasyurids display quite high chromosome conservation. But what you will notice is that we only have very limited Y chromosome information so far for marsupials. And this is because the marsupial Y chromosome is quite small and it's also really heterochromatic. So it's quite difficult to sequence. So we decided to employ a bioinformatic approach to see if we could identify some more Y chromosome scaffolds. We used an average depth ratio approach, which entails aligning male reads and female reads to the male antikinase genome, and then comparing the ratio of the read depth to assign scaffolds to putative chromosomes. So autosomal scaffolds would have a 2:2 ratio. X chromosome scaffolds would have a 2:1 ratio since males only have one X chromosome, and Y chromosome scaffolds would have a 0:1 ratio since the Y is absent in females. Using this approach, we were able to identify 3 scaffolds that we believe to be Y chromosome in the antikinase. And as you can see from this figure, these 3 scaffolds are found to contain majority of the Y chromosome genes that have previously been identified and sequenced in marsupials, which is pretty cool. We then performed some downstream gene family analysis to have a look if anything interesting was going on in the antikinase genome. And we found some interesting expansions in both the Protocadhera GABA gene family and also in the MARK2 gene. So in the Protocadhera gamma gene family, you can see that antikinase have similar numbers to human and mouse, whereas other marsupials have lower numbers of this gene. And the MMRK2 gene is a single copy gene in other organisms, but was found to contain multiple copies in the antikinase genome. And both of these genes have previously been associated with age-related processes and Alzheimer's disease, and so could be interesting targets for future research. Finally, we performed some analysis on some common Alzheimer's-related genes and found that these 6 commonly associated genes were found to be expressed in the antikinase brain, further highlighting the potential for the antikinase to be a disease model for Alzheimer's. So in conclusion, the first antikinase reference genome and associated findings will be a springboard for future research to study age-related neurodegeneration and the genetic interactions between stress, reproduction, and immunity in this Semmelparis marsupial. And I just want to thank everyone who was involved in this project. And we also have a preprint available for this antikinase genome paper while it's in the review process. Thank you.
Thank you, Paris. And next up, we have Andrew Veal from Landcare Research in New Zealand, who will tell us about the stoat genome and how he's using genomics to control pest species in New Zealand.
Tēnā koutou katoa. Good morning, everyone. My name's Andrew Veal, and I'm here to talk about the platinum genome that we've assembled of the stoat, also known as the short-tailed weasel, in order to develop novel tools to control this invasive species in Aotearoa, New Zealand. Now, New Zealand, before humans arrived 800 years ago, was a land without mammals. It was entirely dominated by birds, many of which became flightless and naive to mammalian predation. We brought stoats across from the 1880s and 1890s in order to kill the rabbits, which we'd previously introduced and were destroying the grasslands. Quite rapidly, stoats became a malignant invasive species, predating on the native birds. We have lost about 40% of the birds that were here 800 years ago, and about a third of the birds that still remain are currently endangered, primarily due to predation by invasive species, particularly the stoat in many cases. This means that conservation in New Zealand is trying to kill these invasive species. And there's currently an effort called Predator Free 2050, trying to remove these species from the entire of New Zealand. This requires novel tools, and this is why we sequence the stoat genome. So the Stoat Genome Sequencing Project was a collaboration with the Vertebrate Genome Project. This combines 4 different sequencing technologies: PacBio, 10x, BioNano, and Hi-C. And there are many other talks in this conference on the VGP work. So I strongly recommend that you go to them, and I will not be focusing on this. The Stoat genome is 22 chromosomes, 21 autosomes plus the X and the Y. And our assembly is about 2.4 gigabases. Now, 99.9% of that is in the chromosomes. So it's fully assembled telomere to telomere, full chromosomes. And our BUSCO scores, 96% of genes were complete. So this is a very high quality genome. Now, why did we need to make such a high quality genome? We're doing a number of different things with that, some of which require very high quality genomes. First off, we're creating new forensic markers. This is so that we can identify individuals from trace samples, such as scat, saliva, or hair. So that way we can find, well, how many individuals are in this area? And if we have, say, found a predated kiwi, as is pictured, and we've caught a stoat, is this the stoat that did the predation, or is there another one out there? So this is using new forensic markers created from the stoat genome. We've also been creating a landscape genetics way of looking at things. Basically, stoats are very hard to track across a landscape. You can't put GPSs on them because they're too small. Radio collars, it's very hard to track them because they move up to 65 kilometers in their dispersal. What this means is that it's easier to look at how genes move across a landscape, and then we can use this information to plan control operations so that we can prevent reinvasion to sanctuary areas. Also, knowing the relatedness of individuals is very important, so then we can assess very fine-scale relatedness. Are these siblings, parent, offspring, that sort of thing, and that helps us plan how well an eradication is going. We're also looking at manipulating embryonic diapause, which is something that a number of animals have and is present in stoats. All female stoats are impregnated a couple of weeks after birth, and then the eggs just sit there as blastocysts, fertilized, for about 10 months doing nothing. Now, this is something that we can potentially manipulate once we understand the genes underlying it, so that we can control their reproduction as part of control methods. We're also trying to create new species-specific toxins. Now, this is a receptor that was modeled by my colleague Erica from the stoat genome. So going from genomic sequence to protein structure, and we're comparing those between the different species in that landscape, and hopefully finding some that are different in stoats compared to other things. That means that we can potentially target those receptors with novel ligands or ones that are already known, so that we can create species-specific toxins that do not affect other species in that landscape. And overall, what we're hoping to do is use the stoat genome to help us manage this invasive species and ideally work towards eradication across the country. I'd like to thank a number of people, so all of our collaborators at the VGP, the Wellcome Sanger Institute, and then the funding through Predator Free 2050 and the National Science Challenges. I'd like to thank you all for listening. Cheers, thanks.
Thank you, Andrew. So now we're going to move from invasive species to conservation of endangered animals. I'll just remind you to post your questions online as well, so we have some questions to answer in our Q&A at the end. Our next speaker is Dr. Carolyn Hogg from the University of Sydney. Carolyn is leading an exciting new initiative in Australia called the Threatened Species Initiative, which is one of the BioPlatform's major initiatives. Over to you, Carolyn.
Good afternoon, everyone. So today I'm going to be talking to you about the Threatened Species Initiative. So on the 24th of December, 1968, William Anders took this photo from the Apollo 8 mission, an image that has been described as one of the most influential environmental photos ever taken. To me, it tells a story of technological marvel, of fragility, of hope, and of biodiversity. When Bill Anders took this photo, AI, learning, machine learning, and genome sequencing and the internet were all simply things of science fiction. And what we find now is that in those 50 years since that time, the rate of technology has vastly increased and accelerated. In the same period of time, however, over 330 species have gone extinct, and the rate of extinction is now also accelerating. You just need to look at the media on any given week or monthly basis to see the critical biodiversity crisis we found ourselves in, with up to 1 million species now at risk of extinction. If we look at the IUCN Red List, about 13,500 animals that are listed as conservation dependent or worse, less than 1% of them, in fact, have a genome or genomic resources for them. If we zoom into what we find on the NCBI, we can see that we are getting better at increasing the types of different taxa that we're creating genomes for. But even with all those genomes on NCBI, at least 6% of them are only conservation dependent. And that's in spite of all the literature and recent reviews that have talked about the need and the value for genomes for conservation and for biodiversity. So what's happened in the last 10 years is we've seen a significant decrease in the cost of sequencing around genomes. And subsequent to that, we've seen a massive increase in the amount of genome sequences that we see available to everyone. However, there exists this massive divide. On the one side of the divide are the geneticists and genome biologists creating these amazing genome libraries, whether it be a gold standard, a platinum standard, a diamond standard, a chromosome length assembly, the list goes on. We spend heaps of time perfecting the types of genome sequence that we're creating. On the other side of this massive chasm, however, are the ecologists, the conservation practitioners, and the policymakers who are all pulling their hair out in how do they use this information for their decision making? How do they get better at using the technology that is now available to them? If we really want to go about and lower the extinction rates, we need to get better at bridging this gap. And that's not because managers don't know genetic diversity is important. They know there's a relationship between genetic diversity and inbreeding, and that we need more diversity for future adaptive potential and the fitness of different individuals. And what do we do though with the genomes in conservation? Why aren't we using genomes in conservation? They've traditionally always been considered as a nice to have, and that's simply because of the costs around sequencing and producing the data. If we look here in Australia, we know that of our 200 vertebrate recovery plans, over more than 80% of them have some form of genetic action listed. However, when we delve down into that, less than 15% of them are using genetic data to make their decisions in their recovery actions. When we talk about genomics and management, we really are talking about having a reference genome, either for the species of interest or the cogeneric species. And the reason we're looking for these reference genomes is because it allows us to do functional gene characterization, whether it be immune genes for species suffering from disease, reproductive genes for those that we know there are key reproductive issues, behavior genes, or even heat tolerance genes to have a better understanding of how species will potentially adapt to the changing climate. The more traditional way we've been using genomes in conservation, of course, is with SNP-based technology, where people are using the SNPs to discover and discuss population differentiation, population genetic diversity, and inbreeding effects. But why is it so critically important for us now? Well, the bushfires of the past season have significantly highlighted for us, with the 1 billion animals lost and 23 million acres burnt, what a critical state we now find ourselves in. The reason for that particularly is in Australia, over 8,000 of our vertebrates are endemic to this country alone. And so in a recent publication that came out in Nature, Ecology and Evolution, where they investigated the fauna impacted by the recent bushfires, we can see that the number of species that are critically endangered lost large proportions of their habitat. And so the way of dealing with this situation is to revegetate this habitat, translocate individuals, perhaps bring them in for a captive breeding program, and try to figure out ways to ensure that they can remain in the landscape. So the Threatened Species Initiative is basically designed to be built on the back of the Bioplatforms investment into the other genome initiatives in Australia. But unlike the other initiatives, it's not about just creating genomic resources for our threatened species or associated population data. More importantly, the list of species that we've created for the Threatened Species Initiative is really being driven by the conservation sector. What do the state government agencies require? What do the NGOs require? What do the managers need on the ground to make better decisions? Another key aspect of the Threatened Species Initiative is to develop an online conservation portal so that conservation managers can start to take genetics into their own hands, be able to use the information and apply it in their real-world decision-making rather than always having to wait for the academics or the researchers to come to them with the answers. So how does it work? Well, we know that we can create reference genomes and assemble and annotate them through the TSI and also the other bioplatforms projects. These are available along with the other suite of other genome initiatives around the globe. So the simple case is the end user collects the sample themselves. They can send it off for the commercial sequence provider. They receive the data back, and then they can go to this online portal. The online portal will allow them to load up their data, will be able to hover over things, answer questions, decide what kind of assembly alignment method they want to use. They'll also be able to input their reduced representation sequencing data is where we're going to start. They can select their downstream analysis that they want. They may be able to affect some of their advanced parameters. They submit the details and they get a submission summary. And what happens at this point, it will fire up a cloud computer, will run the analysis for them, and then they receive the data back in a really easy interpretable format. And with they can have that standardized report, they can understand what the questions are being answered by their data set. Are my two populations different? Should I be mixing these animals? Are my founders in my captive program related to one another? And then they can use this information in the decision-making process and the cycle begins again. Well, this is currently what we do with the Tasmanian Devil program and some of the other species we work with here in Australia. And if you look at it, we can collect the samples in January. It goes through the whole process and we're helping the managers make end user decisions in about 4.5 months. So the Threatened Species Initiative has actually started. We've selected a number of pilot project species. And the reason we've chosen these species for the pilot project is really key to answering some aspects of the workflow. So we're trying to refine all the different parts of the workflow before we go out for a larger expression of interest round in 2021 and hopefully be able to create genomic resources for about 50 of our threatened species. It's by no means a small project. A large number of people are involved in this from all the from a number of universities, zoos, but most particularly the federal and state government agencies across the country. So for us, really, it's about having the geneticists and genome biologists on one side and integrating with the conservation managers and policymakers and really designing a system that can bridge this gap so we can help use better the genomic resources that are available to help the species at hand. So the question I come back to you all on this is over 50 years ago, they took this photo and we know that we've made massive technological advances. We can now use this information to slow the rate of extinction. And so the question I leave all of you with are, if you're creating genomes, how are you creating them so conservationists can use them? How is the information that you're creating actually going to have an impact? And most importantly, what will our planet look like 50 years from now? Thank you.
Thank you, Carolyn. Our next speaker is Professor Arthur Georges from the University of Canberra. Arthur is leading a new bioplatforms initiative, AUSARG, which covers amphibian and reptile genomes from Australian animals. Over to you, Arthur.
I'm Arthur Georges from the University of Canberra, and I'm going to present on AUSARG, which is an initiative funded by Bioplatforms Australia on Australian reptile and amphibian genomics. Most of you would be aware that Australia is an island continent and it's been isolated for a long time, and so there's a high degree of endemicity in the fauna and some really to contribute genomics in a very special way. The context of these radiations include long-standing isolation, so they've had time to radiate, but progressive aridity has been an important component of the equation. All of Australia's rivers at the moment, if you add up their discharge, comes to about the same discharge as the Missouri River into the Mississippi. So it's the driest vegetated continent on Earth, and that has a really profound impact on the biota. And the climate's really unpredictable. So it's a land of droughts and flooding rains, as they say. And of course, this all provides unparalleled and unique opportunities for studying the biota in ways that can't be done on other continents. And we put this proposition to Bioplatforms Australia, and they came to the party and funded the AusArg initiative to bring genomics to bear on not only accelerating discovery in our unique continent, but also in making tractable questions that were previously intractable because of the lack of genomic information. So we consulted widely with the herpetological community and came up with some criteria that would enable us to select the 15 to 25 genomes that we were going to work on. And these are the core criteria. The first thing is that the genome needs to be relevant to questions of substance in either conservation, management, or discovery more broadly, and likely to be published in leading journals. So we're not just producing genomes for the sake of producing genomes. We want some companion outcomes. The team's really important. We have to have a winning team that has a capacity to assemble, annotate, curate, and physically map and communicate the draft genome assembly, all using the latest technologies, and to deliver on the companion studies. And next, there needs to be resources. So the team has to have the capacity to marshal the funding necessary to complement the Bioplatforms Australia contribution. So the team has to be Australian Research Council competitive, basically. And finally, in terms of the core criteria, they need to have access to the tissues preserved in a manner suitable for extracting high molecular weight DNA for long read or ultra long read technologies. So they're the core criteria for selecting the projects or the genomes that we're going to proceed with. There are some other criteria. The genome size has to be tractable, so that it can be sequenced, assembled, and annotated at a reasonable cost. The taxon or the species needs to be commonly available, so that once the genome's generated, lots of people can work on the species with the new genomic information, and it needs to be amenable to captivity potentially. These aren't essential criteria, but they help move a species up the list. So if they're amenable to captivity, it means that people can work on them in laboratories and conduct their research in a context of having a readily available model species in the laboratory. And finally, it's great if they're distinct. So we want to get coverage across the major clades across the Australian biota. So here's some of the projects first off the rank. First one is looking at the evolution of viviparity and placentation using Pseudomonas and Bassiana and Lampripholus, and Oliver Griffith is the project lead on that. Second project's on limb loss, looking at Scyphus and Larista, and Camilla Whittington is the Australian taking the lead on that, but she's collaborating with Darren Card and Scott Edwards in the US. I'll be looking at sex determination with Claire Hollerley in Bassiana and Pagona and related species to see if we can tease out the sex chromosomes using the phasing capabilities of the new technologies. Craig Moritz and Janine Deacon and Tony Gamble will be looking at chromosome evolution in geckos. Mike Gardner and Terry Bertozzi will be looking at host-parasite interactions in Tiliqua. And we've got some frogs in there. We're looking at the evolution of genome size in platyplectrum, and Sangeet and Renee Catullo will be looking at that, along with a bunch of collaborators. And of course, we wouldn't want to miss out on the Elapidae. So Katie Sanders is looking at marine transitions and what the elapids have done in order to be able to move into marine environments. And that's being done in concert with Rich Edwards and Vicky Thompson, who'll be looking at brown snakes, tiger snakes, and perhaps a taipan. Okay, so they're the sorts of projects that we're running with because they meet the criteria. There's a bunch of other species that we'd like to run with for want of a champion. If there's no champion, these things won't run. Our strategy is to use either PacBio Hi-Fi plus Hi-C or ONT Prometheon plus Illumina short reads for polishing plus Hi-C, and that's a decision for the project leads. Of course, I guess what legacy data they might have already might govern this decision. If they've got a lot of Illumina short reads, they might go with ONT Prometheon. We're well aware of the fact that sample quality is really important, and we're recommending snap frozen blood. Reptiles and amphibians have nucleated blood cells, though it seems that pretty much any tissue, provided that it's been snap frozen, is suitable to generate the high molecular weight DNA that's necessary for these long read technologies. We've trialled spooling, circulomics kits and LSK 110 ONT kits for extracting high molecular DNA, and they all pretty much give comparable results. The spooling is obviously a lot cheaper, and you get an awful lot of high molecular weight DNA. Here's an example of where we applied the spooling technique for Pagona and ran on a femtopulse, and we've got obviously high molecular weight DNA, 165,000 base pairs for the peak there is really good. When we ran it on a low output flow cell, we got really good results from the ONT low output flow cell using the spooled high molecular weight DNA. We also had a look at this FedEx sonication issue, and it appears that it's not a big problem in Australia, relatively short distances for the transport. It didn't really matter whether we shipped the DNA unfrozen or frozen on dry ice. Basically, the results were fairly comparable in terms of the outputs from the ONT. So the FedEx sonication doesn't seem to be a big problem for us in Australia, though I guess if you were sending it from Australia to the US, it might be. And then finally, we've got some early results. So for Tiliqua on the ONT, some spectacular results. The Contig N50 was 11 megabases. We got read N50s of 35 kb, and the longest read was 757 kb. So really good results for the sleepy lizard. Not so good for the dragon. High heterozygosity might be an issue here, but the Contig N50 was 690 kb. And I think the longest read was about 505 kb. So these are really encouraging results, and coupled with the Hi-C, we believe we're going to get really high quality genome assemblies and annotations from these species. So in conclusion, I guess it's early days for this project. We've only been running a few months, but really exciting times. We're hoping to generate some great genomes from some great Australian species. But of course, there's too many species and too little time. So there's an open invitation for anyone who wants to partner in generating genomes for some of the Australian species that aren't necessarily going to be part of the 15 to 25 genomes of Bioplatforms Australia. So yeah, partner is the is today's motive. And I'll finish up just by introducing the people who are in the team. So AusArg comprises comparative genomics with myself and Renee Catullo leading that, a taxonomy and conservation program with Jane Melville and Jody Rowley leading that, phylogenomics program with Craig Moritz and Scott Keogh leading that. And of course, Bioplatforms Australia is there in support with the CEO, Andrew Gilbert, Sophie Mazard, who's been allocated to AusArg, and Johan Gustafsson. And hopefully we'll get to do the genomes of these two spectacular species as well. And I'll leave it there. Thank you very much.
Thank you, Arthur. The last of our 6 speakers is Professor Peter Dearden from the University of Otago. Peter's going to tell us about building genomics capability in Aotearoa.
Hi, my name is Peter Dearden. I'm from the University of Otago in New Zealand and also Director of Genomics Aotearoa. I'd like to thank the organisers for allowing me to speak to you today. I want to talk a little bit about New Zealand and how we do genomics in New Zealand, and maybe tell you some things that might be useful to you. One of the cool things about working and living in New Zealand is New Zealand is a very unique place. It's biologically unique. The organisms that live here, many of them live nowhere else. Many of them have strange and unusual adaptations to strange and unusual environments. Same applies to primary production systems. It turns out when people arrived in New Zealand, they discovered that you couldn't make food in the ways you would in other parts of the world. And so our primary production systems are also unique. And of course, finally, our people are unique. New Zealand was originally settled by an indigenous group, the Maori, and the state of New Zealand was founded on a treaty between those indigenous Maori people and the British Crown. That treaty is of vital importance in New Zealand today, and it obliges the New Zealand government and its agents, including researchers, to partnership in the exercise of governance. This partnership model really hasn't been applied well in the past. Many of the genome sequences from New Zealand's iconic organisms have been carried out overseas from specimens in people's freezers or museum specimens. And what that's meant is that we've kind of disconnected the development of a genome sequence from that organism and the need to conserve and look after that organism. So other people's research priorities have driven genomic research, and New Zealand organisms have to some extent been strip mined by oversized scientists. What this means is that we can't use some of that information to help us better manage the species that we have, and we haven't been able to use those species that we do have to build capability and capacity amongst New Zealand scientists or among New Zealand Maori. So one of the solutions to this, and what we're trying to work on now, is this grouping of Genomics Aotearoa. This is a national platform of universities and Crown Research Institutes set up to benefit New Zealand biogenomics. It's supported by the Crown, so we're funded by the Ministry of Business, Innovation and Employment. We have 9 partners and 30 associate organisations, all of whom are interested in developing and ensuring that we have the capability and the capacity to do our own genomics research and ensure that genomic research carried out on New Zealand organisms really benefits those organisms and New Zealand. One of the most crucial aspects of this is that, going back to that partnership with Maori. Maori have very clear views about the organisms that live in the New Zealand environment, and many of them are known as taonga. Taonga could be loosely translated as treasures. Taonga are very important to Maori because they are an insertion of another Maori term, kaitiakitanga, which really means governance or rights and responsibilities to an organism. They're also an acknowledgement of mana, of respect for a particular group of people. So taonga are deeply woven into the way Maori think about their environment. All endemic species in New Zealand are taonga because the ecosystems in New Zealand are taonga, they're treasured, unique environments. Also, introduced species that are important food sources, for example, can also be taonga. But perhaps most important for genomics, the species, the genome sequence derived from it, the bioactive compounds derived from it, associated microbiome, they are all considered taonga. They're all part of that organism. And so when we talk about governance, rights and responsibilities, kaitiakitanga, this applies to the genome sequence as much as it applies to the organism. And this is kind of challenging because Maori communities want to have ongoing guardianship of their taonga, including genome sequences. They wish to have the right to decide how genome sequences are used and who uses them. And that isn't really doesn't align well with genomic science's need for open access. So one of Genomics Aotearoa's big roles is trying to reconcile guardianship and access. And that's problematic, and we're working on it, but we're hoping to find solutions whereby actually the guardianship of Indigenous groups can be respected while access can be allowed. Because of this, it means that I'm not going to stand here and give you a list of species that we're sequencing the genomes of, because actually many of those are in a discussion phase with Maori, and we can't prejudge whether Maori groups will say yes or no, or whether how they want to be involved, or whether we should sequence something else. So I don't really want to give you a list. I'll give you some examples of things that we have sequenced already, and really where we're moving to. So we started genome sort of sequencing work really focusing on high quality genomes of particular species. Here's 3 examples. We've generated high quality genomes. We have a lot of bird fauna in New Zealand. This one here, the hihi, we've generated ways of producing very high quality chromosome level assemblies of these kinds of birds. We have lots of unique plants. This is the rewa rewa. Again, we're trying to develop ways to ensure that we can produce very high quality genome sequences to support both honey production, it's an important plant for honey production, but also conservation efforts. We're also grappling with huge genomes. This is my favorite invertebrate, the velvet worm, or an onychophoran. This has a 12 gig genome, and we're finding ways to sequence that genome, assembling, stitching together with long reads, etc. It's scientifically important. We also focus on species of importance to New Zealand that aren't natives. So we've sequenced the genomes of Vespered wasps, for example, which are invasive and damaging in New Zealand environments as a way to get at new ways of controlling them. We're sequencing the genomes of parasitoid wasps, which control pasture pests, and of course the mammalian predators that have been introduced to New Zealand and damage our environment and our conservation species like the black rat and the possum. But we sort of moved beyond that generating a single genome of a species, mainly because of this animal here. This is kakapo, a fantastic fat flightless parrot that lives nowhere else in the world, and of which there are only about 200 left in the world. This organism, the genome sequence has been available for some time, but we now have a genome sequence of almost every living individual, which allows us to move from the genome sequence and understanding something about the evolution of the species to start understanding population structure. This is an example of a single kakapo chromosome where we've taken the whole population and shown genes and transposed elements, for example, but also runs of homozygosity, recombination in a kakapo pedigree, local population structure, etc. This sort of information is exactly what we need to feed back to our conservationists to ensure that we are doing our best to improve the future for these rare and endangered animals. We're doing the same sort of work for species which are perhaps more mundane, but things like honeybees in New Zealand are underpin an extremely important part of our economy, and yet we have worries with inbreeding, and our bee populations are just different from many in the rest of the world because they come from admixtures of different groups of bees, and this leads to weird situations. Here we have inversions at very high rates in New Zealand bee populations, which have implications for management. So moving beyond genome sequence to population genomics is where we're going, but we're doing this in a way that enables us to work with our own native species and ensure that we're benefiting their conservation. So this is a model I think we're thinking about for other countries as well, focusing on research priorities to benefit New Zealand, which includes management of our critically endangered species. This is beyond just sequence. It can't just be genome sequence. We have to move to population genomics and using that population genomics to better understand the dynamics of those populations. We're also supporting Indigenous aspirations by getting Indigenous voices into research conversations, ensuring that they are engaged with this research and that they are building their own capability and capacity to do the research themselves. We're also very keen on explicitly building capacity for the future by supporting careers, ensuring that people who work for us work on interesting projects, get high impact papers, and therefore support an ongoing future in this career in New Zealand, thereby ensuring that we can do this work into the future. That's all I have to say, except to thank all the people in Genomics Aotearoa who are awesome, and some of whom are speaking today as well, and the Ministry of Business, Innovation and Employment from the New Zealand Government who fund this work. Thank you so much for your time, and thank you for allowing me to speak.
So, Andrew, I'll start with you. One of the questions is, could you comment on possible unwanted consequences of the elimination of all mammal predators, considering their ecological roles?
Well, it's a good question, and there are potential side effects of removing predators. However, if we can remove all of them, in all the situations that have occurred, generally on islands where we've eradicated using Britificum, all the invertebrates and birds have returned to what we assume to be far more normal levels and recovered. So there probably aren't that many negative effects removing them all. Removing some but not others has the potential for mesopredator release. And that's something that we are still looking at in terms of, well, what order is best to do things. It might be that if we remove stoats, weasels increase, and then invertebrates or reptiles decrease. So it is something that's very important to look at while we consider trying to get rid of all of these things.
And that leads into another question, which is, given that all mammals are pests in New Zealand, why not use a mammal-specific toxin to eradicate them?
Not all mammals are pests, in that humans are mammals. We also do have 2 native bats, and we have sheep and cattle and dogs. And so it's far better to have things that are specific to the pests, but we are looking for ones potentially for, say, carnivore-specific toxins. But yeah, it's nicest if we can actually target things quite specifically. And also, one issue, as you may know, is that we use 1080, which is a toxin which is relatively broad spectrum across mammals and birds. But for that one, deer are a pest, but they're also a resource. And it isn't a good death for those. So having things that are very specific enables us to do different things in different areas and avoid potential conflict and problems.
And one technical question, what sort of genetic variation did you look at for the stoat forensics project? Okay,
that wasn't mentioned in the talk. So the genome was made from one male from a relatively inbred population, which means that it assembled really nicely. However, we have done GBS on about 120 individuals from across the country, and then looked at variation across the entire country, trying to choose those SNPs so they're at least 1 million base pairs apart. So there shouldn't be in any linkage with each other. They're highly heterozygous across the entire country. So that was the population level stuff for the forensic markers. And we will be doing some low coverage genomes as well.
Great. So Carolyn, over to you. There are a couple of questions for you. So firstly, what's the awareness level of the importance of genome data in Australia among the conservation practitioners? I'll stop there.
Yeah. All right, just looked like you were going to keep going. It's actually very high. So when we first started discussions about whether or not we were going to start the Threatened Species Initiative about 18 months ago, we did a lot of consultation around the country with different threatened species managers. Within Australia itself, we have states and territories, and each state and territory has a threatened species manager that is responsible for the management of those species within their state or territory as a government agency. And so we met as a collective group with all those agencies, and then did kind of like a roadshow going around the country last year, or just via Zoom calls, meeting with all those different agencies to have conversations with them about the value of having genetic information in their decision making. And pretty much from all the government agencies, the zoos, as well as the NGOs that run the large fenced enclosures here in Australia, was pretty much collectively, they all recognise that it's important. But the critical thing for them is they don't know what they're supposed to, how they're supposed to access the information, and then how they're supposed to utilise that information. So there's this really big disjunct between what we produce as genome biologists and what conservation managers actually need. And that's what TSI is trying to bridge.
So another question is, can TSI be used by countries outside of Australia? And if so, what's the data ownership policy?
So at the moment, no, because we're still in the pilot phase. We're just doing our pilot project at the moment, and we've just started the development for the web portal. My global ambition is I'd like to build we'd like to build this conservation toolkit platform that can be used anywhere in the world with any kind of data set. So that's pretty ambitious, but I always think if you don't shoot for the moon, you're never going to get there. So we're going to try and shoot for the moon twice over in this project. But with the data, you're not actually loading your data up for permanent kind of residency or storage within the portal. The portal is basically a way of firing up a supercomputer for yourself in the cloud and being able to run your analysis quite rapidly, because anyone who uses genomic data can tell you it's big data sets, and we have to work out ways that we can process that information quite quickly. And a lot of conservation managers who work for government agencies or NGOs just don't have access to that kind of high performance computing that you do if you work at an academic institution. So we're trying to build something that gives them that same access in the cloud system. And then basically once they receive their data back, they just shut down the computer. And as you shut the computer down, it just deletes everything. So that there shouldn't be any IP issues, hopefully.
Excellent. And I think you've touched on this with this next question. What's needed to improve global integration and collaboration of in situ and ex situ genome-based conservation programs?
I think as geneticists and genome biologists, we have to stop using so much jargon and learn to distill down the technicalities of genetics and genomics into such a format that people who are not experts in this area can understand what you mean. And I think if we get better at that communication side of things, my hope is that we'll be able to integrate it more easily across all different types of management globally.
And finally, to what extent are you worried about certain aspects of a standard pipeline? For example, Hardy-Weinberg filtering across diverging populations?
Yeah, pretty worried. This is a discussion we've been having quite significantly within the working group that's around the platform. So we've just started a small team at the moment, and the idea is to trial it with some projects and then find out where the kinks are. And then as we get it starting to work, we then would like to bring on other people who work in this space as well and grow it iteratively. So I don't think we can create anything from the start that will answer all questions immediately. The plan is to simply start with something that answers the most commonly asked management questions and then build it from there.
Fantastic, thank you. Now over to you, Peter. Could you elaborate more on how you reconcile guardianship and need for open access in genome science?
Yeah, so this is a difficult question and an ongoing conversation. So, you know, the need to ensure that that the people who have guardianship over species data actually have a meaningful role in allowing or not allowing access to that data really does butt up against the idea in modern genomics, which, you know, I support, which is that sequences should be open access. And the problem here is one where those two ideas are kind of not complementary, and so we have to find some sort of mechanism where we can allow guardianship to happen while access also occurs. And so currently Genomics Aotearoa has a data repository where if you ask for a data set from us, then we go and find ways to ask the people from where that genome came from. And that's one approach. I think it's a really difficult approach, and obviously, if we're talking about the thousands of times the genome might be accessed in a year, that's really going to be clumsy and hard to implement in the long run. So we're also thinking about are there ways that genomes or sequence data could be labeled in international databases as coming from an indigenous source and saying this particular genome sequence is not for commercial use, for example. Such a thing works really well with kind of Creative Commons copyright labels. So I don't see why that can't work in a community that really values access to data. It would just mean that we would have to regulate ourselves and be able to say, yeah, actually, there are some constraints on the use of this data, and those constraints are reasonable and not something that we can just ignore.
Thank you. And also, can you talk a bit about how you're building capacity within Indigenous people in genomics research in New Zealand?
Yeah, sure. And I think Caroline actually pointed out some of the key things here. this is not an unusual problem, and it's not a problem with just Indigenous people. It's about how we best communicate the science and the utility of the science that we do, and ensure that communities, whether Indigenous or not, understand what that science is capable of doing and can interact in such a way that they say, these are things we want to know, and this is, we would like these sorts of things to be investigated. So there's a bit of that talking with communities, particularly when we're sort of asking permission to work on a particular species. There's also a real role of saying, this is what we can do with this technology, and what are you interested in? What could we do together? What is it that you want to find out? And how can we bring new people into this kind of science? We also run an internship program over summers in New Zealand where Indigenous people get together and talk not just about the interface between sort of science and what they do, but also how giving them the opportunity to talk about how genomics should work in a Maori worldview, in Te Ao Maori. And I think that's a really useful thing. So this is not a deficit model. We need to show you how this works and you need to interact with it. It's more saying, look, here are the tools. How would you like to use them? And I think that's really critical. And of course, we bring lots of, well, there are excellent Indigenous scientists in New Zealand working in genomics, and we support their research and try and support their ambitions and to ensure that we're training more and more students. So it's a long-term process, and I think that's the critical thing. You can't do this overnight. You have to have long-term engagement with community, long-term pathway and capability building. And only when you do that are you really going to have an ecosystem where Indigenous groups have real participation in the science and are not researched on. They are the researchers and they ask the questions.
Thanks, Peter. And Arthur, I'm going to ask you a question so that you don't miss out. I'm really curious to know, the AusArg initiative, studying Australian amphibians and reptiles is very exciting. You've got a lot of programs going on looking at things from the evolutionary history and the radiation of the reptiles, viviparity, placentation, limb loss, sex determination, chromosome evolution, host-parasite interaction. There's a lot of programs going on there. How are you empowering the herpetologists to work with the genome scientists, to really, deliver on these programs? Because many of the herpetologists that I do know haven't necessarily had a lot of genomics expertise. So how do you build that community when you're starting a new project like this?
Yes, we set up a bioinformatics expert group, and so I wrote to people all over Australia to try and see if they were interested in providing advice to the community who were interested in generating the genomes. And as it turns out, everyone I approached said yes. And so we've got a really sterling group of individuals who have the expertise to be able to pull together the genomes, either providing advice on sample extraction or providing advice on assembly or providing advice on annotation. There's a whole, a full range of expertise that's brought together, and that panel looks at the proposals that are put forward and provides advice back to the project team as to how they might best address their questions using the different platforms that are available. And so it's really important that the question drives the platform that's available. We're not trying to produce platinum genomes for every species we look at. We look at the questions, and then the bioinformatics expert panel advises back onto as to what the best platforms are to get the best bang for the buck and answer their questions. So, and that seems to be working really well.
Fantastic. Well, thank you to all of our speakers, including Anne and Paris, who weren't able to join us for the session today, but thank you all. And thank you to those of you who listened to our Q&A through the recording. I hope you enjoy the rest of the meeting. Bye-bye.